Good day to all of you. This video is about the receptors and their types. So receptors are the proteins associated with the cell membrane are located within the cells, and it recognizes signaling molecule by binding to them. Cell surface receptor categories can be separated into four classes, that is G protein linked receptor, ion channel receptor, receptor lacking intrinsic catalytic activity but directly associated with the cytosolic protein tyrosine kinase, and fourth one is the receptor with intrinsic enzymatic activities. So first one is the G protein coupled receptor, also known as the GPCRS, and it includes the epinephrine, glucagon, serotonin. So in this figure, it is the plasma membrane, it is the cytosolic part, and it is the exterior part. R is the receptor protein, and G is the G protein, which is an inactive stage, and is the effector protein. When the ligand is added on the receptor, the G protein is activated, and then reaction proceed, and then it bind with the secondary effector cells to generate the secondary messenger. In the next part, the non-catalytic tyrosine kinase linked receptor. In this figure, when ligand is added on the receptor, the inactive tyrosine protein get phosphorylated, and with the help of the ATP, the substrate protein get phosphorylated. So these next one is the intracellular receptor. So this figure depicts that the cell surface receptor, and it is the intracellular receptor. For intracellular receptor, for lipid molecules may be the transcription factor enhancing or the suppressing gene expression. The interaction between the ligand and the receptor causes the generation of the second messenger. So when it is a cell surface receptor, and when ligand is added, it is called as the first messenger, and then it comes into the cell, then the second messenger intracellular effect, and then when it enters into the nucleus, it is also known as the secondary messenger. Example of secondary messenger like calcium camodulin, cyclic AMP is very common. And second messenger are molecules that relies signals from receptor on the cell surface to target molecule inside the cell. Second messenger are short-lived intracellular signaling molecule. Elevated concentration of secondary messenger leads to rapid alteration in the activity of one or more cellular enzymes. There are four classes of the secondary messenger like cyclic nucleotides, membrane lipid derivatives, calcium and nitric oxide or carbon monoxide. Types of secondary messenger, like it may be the hydrophobic, hydrophilic gases. Examples are here. Cyclic AMP, it is a secondary messenger that is synthesized from the ATP by the action of the enzyme adenylyl cyclase. Binding of these protein to its receptor activates a G protein, which in turn activates the adenylyl cyclase, leads to the appropriate response in the cell by either or both. Example of cyclic AMP like adrenaline, glucagon, LH. Mechanism of receptor mediated activation and inhibition of the cyclic AMP. The ligand binds to receptor, altering its conformation and increasing its affinity for the G protein to which it's bind. The alpha subunit dissociate from the G complex, bind to an effector in this adenylyl cyclase, activating the activator, and activated adenylyl cyclase produces cyclic AMP. In this figure, it is a receptor, and it is the G protein bound to the GDP. When the ligand are added in it, the GTP is converted into GDP and the activation of the G protein, and then G protein the breaks into smaller subunits alpha and beta and the gamma, and then the reaction proceeds, and then the GDP through the by the help of the 1-phosphate, the alpha subunit of the G protein attached to the effector cells, and then it get phosphorylated, and then the effector cells, the arrestin is added on in it, and then GDP is further unite to form the G protein. So the next step is the receptor has been phosphorylated by our GAR key. This phosphorylated receptor has been bound by an arrestin molecule, which inhibits the ligand bound receptor from activating additional G protein. So glucose mobilization induced by the cMC cyclic AMP. So in this, there is a capillary, the peptide hormone, which act as a first messenger, and then the activated enzyme, it converted into the ATP to cyclic AMP, and the receptor protein binds to the peptide hormone, and then the ligands are attached on it. Here is a descriptive steps of hormone bind to an receptor into the plasma membrane, then binding leads to an activation of enzyme that changes ATP to cAMP, and then The phosphorylase kinase phosphorylates glucagon by phosphorylase, stimulates glucagon breakdown, glycogen synthetase inhibition prevents conversion of glucose to glycogen. It is also a cyclic GMP. It is synthesized from the nucleotide GTP using the enzyme guanylyl cyclase. Many cells contain a cGMP stimulate protein kinase that contains both catalytic and the regulatory subunits. cGMP serves as a secondary messenger for the nitric oxide response of the rods of the retina to the lights. Inositol triphosphate is a soluble molecule diffuses through the cytosol and binds to a receptor on the endoplasmic reticulum, causing the release of calcium into the cytosol. So this is all about the receptor, the G protein cyclic AMP and the regulation. So thank you and keep watching.